Hi guys, welcome back to Biology Videos at TDA. In this lesson we're going to look at voluntary and involuntary muscle of mammals. So by the end of the lesson what we should be able to do is understand the structure and types of mammalian muscle, and then be able to interpret diagrams of photomicrographs, and then be able to fully label something called a sarcomere. So we're going to start off with a bit of an introduction on different types of muscles. So there's three main types of muscles that we find within mammals. We're going to see in humans for the rest of this lesson, but it's the mammals, which is cardiac muscle, smooth muscle, and skeletal muscle. And we're going to look at what each type is used for. Muscle takes up a huge amount of our body mass, particularly the more muscular you become. But on top of that, you have a lot of smooth muscle inside your body that you're probably less aware of, that takes up a lot of mass and space. So the first type of muscle, the one you're most familiar with, is skeletal muscle, and that's the muscle of the skeleton. So your biceps, triceps, quadriceps, what you know of as your external muscles on your body that you use for exercise and use for movement. And they're going to be situated in antagonistic pairs. That means there's two pairs that work together, such as the bicep and the tricep. The bicep's going to cause flexion at the arm, and the tricep's going to cause extension at the arm, so at the elbow joint. And this is almost entirely voluntary, so you can control these muscles consciously. You may do things autonomically, you may do things without thinking, you may have reflex actions that are involuntary, but we refer to skeletal muscle as voluntary muscle. Then that takes us on to cardiac muscle. So heart muscle, it's the only place we have it. We'll look at the structure of it a little bit shortly, but the key first point to note about cardiac muscle is it's myogenic. So that means it contracts on its own without help from the nervous system. Now we have control of the heart. It doesn't control itself completely. It has its own pacemaker, the sinoatrial node, if you remember from your GCSE, around here. And it does control its own rhythm, but then that's influenced by the brain itself. So they'll have a sympathetic and a parasympathetic, so the sympathetic nerve and the vagus nerve, which is the parasympathetic nerve, which will increase and decrease the rate of that. So it's myogenic because it can contract completely on its own. And then that takes us to smooth muscle. So smooth muscle, the muscles inside our body that move things inside our body. It's generally involuntary, so we don't have to consciously think about it, but with training, and there's some examples that can be both, such as the diaphragm's a bit of an exception. It is involuntary, and it's referred to as skeletal muscle technically. But that's involuntary most of the time. But generally, smooth muscle is going to be things like muscles of our intestines and sphincters, and muscles that move food around in our body, or muscles within our arteries and veins. You may remember we have some muscle in there that can contract, and you don't think about the shunting of your blood. It's done automatically due to hormones. So we're now going to look at photomicrographs and images of muscle. So you need to be able to, from an image, or I'll find some exam questions that are similar to this, be able to identify the type of muscle you have by the image. So if I just go to a diagram to start with, and if we show the labels, you can see from the structure of cardiac muscle, it's not very uniform. There's these kind of layers that we can see of the cells, but it's not perfectly segmented like skeletal muscle is. A couple of key points to know that it's cardiac muscle is the first one is that it's mononucleated, so it means that there's one nucleus per cell generally, and it also has these intercalated discs. These little discs, they help the contractions between cells and they go between them, and it's also still slightly striated. So when we go to the photomicrograph, we have these striations. So that means these bands of different colours, and we'll look at that when we get to skeletal muscle. So skeletal muscle is much clearer and obvious to see, and if you look at the difference between the two, the staining is different slightly. We've got a darker stain, but you can see these really uniform striations with skeletal muscles, so they're all really similar. There aren't these kind of different funny shapes. It's really nicely uniform. It's also multinucleated, so it means it has more than one nucleus per cell, and we'll look at this muscle in a bit more detail in a second. The key thing about skeletal muscle is it's really obvious that you have these light and these dark sections, these striations, and we'll come to that a little bit more. And then finally that brings us to smooth muscle, and it's much easier to spot compared to the other two because it's unstriated. They aren't these nice bands, and there's a lot less myosin, and we'll come to that. They're also mononucleated as well, so you can distinguish them. It might be worth drawing a small table now and then listing that so you're really clear on that. And you see from a diagram it's not as clear. So we'll look at skeletal muscle now, and we'll look at the different types of skeletal muscle. So there's lots of different versions of this, and I did some research a few years ago, and I found 16 different types of muscle fibres. So there's lots and lots of different types, slightly different versions, but there's two main types: slow twitch and fast twitch fibres. And the way we think about it is fast twitch fibres contract faster, they're more powerful, so therefore it's more anaerobically, so they're going to be using phosphocreatine. Now phosphocreatine is essentially a store of ATP. If I just go back and show you this slide. So phosphocreatine is this compound called creatine that has a phosphate group attached to it, and the way it works is it then donates its phosphate group to ADP, adenosine diphosphate, to make adenosine triphosphate, and that leaves creatine left over, and that then can be used for really rapid energy in things like fast twitch fibres. That's why you might see bodybuilders and people that do exercise, they take the supplement creatine because they think it increases the store. Whether it does or not, I couldn't tell you for certain. But they think that there's an increase in creatine stores in the body if you take supplements of creatine. But that's why, because it's essentially a phosphate donator to the ADP once you've used up your ATP, and then you can kind of recycle it. So these fast twitch fibres are going to be using up this phosphocreatine along with ATP mostly, and they're going to produce in lactic acid lactate. So that's fast twitch. Slow twitch is much more slower. That's going to be done aerobically. As well as that, in slow twitch fibres, there's a couple of differences between them. They have a lot of mitochondria. Now that makes sense if slow twitch fibres are working aerobically, so they require oxygen. That's going to be used in aerobic respiration from mitochondria. They also have a high concentration of myoglobin, which is very similar to hemoglobin. Imagine it's the same thing as hemoglobin but in muscle. So it absorbs oxygen, it has a high affinity for oxygen, so it will take oxygen from hemoglobin into the myoglobin, which goes into the muscles, which can then be used. Also, slow twitch fibres, they will be using ATP because that's produced by aerobic respiration, but they use it much more slowly, but then they fatigue, they tire much more slowly. There's some belief that certain athletes are born, and we're all born with different distributions of slow twitch fibres and fast twitch fibres, which is why some people are just naturally faster at running or sprinting or more powerful. But these can change. You can train your body to go more into fast twitch and more into slow twitch. But there is a good set of evidence that we are born with different combinations of this, but don't use that as a reason why you're not very fast. You can train if you want to run faster and that kind of thing. Okay, so we'll have a go at some questions. So question 1. Which of these fibres tend to respire aerobically? You can just say it out loud, think it in your head, or write it down. So aerobically is going to be slow twitch. Next one. Which of them become fatigued quickly? It's going to be fast twitch. Next one. These fibres produce more powerful contractions than the other one. It's going to be fast twitch again. Next one. Which one is rich in myoglobin? Slow twitch. And which ones contract slowly? Slow twitch. Which one are rich in mitochondria? Slow twitch. These fibres use large amounts of phosphocreatine to regenerate ATP. Fast twitch. These fibres have an excellent blood supply. Which one do you think would need excellent blood supply? I didn't mention it before. Which one needs a lot of oxygen? Slow twitch. Which one are known as white fibres? Or see if you can apply thought to this. They'll be fast twitch because they have a lot less myoglobin with them and they have a worse blood supply. Okay, so I'm going to give you an idea about what a sarcomere is, but first of all I'll just kind of give you an overall introduction of the muscle fibres itself. So muscle fibre is like a piece of rope. It's bound, lots of these fibres bound together, and this is essentially one cell, and the sarcolemma is the plasma membrane which goes around the outside of the cell, and within inside the cell it has these muscle fibres in them. Now you can see these blue things here, these blue and purple things on the outside here. And this is trying to represent something called T-tubules, which are essentially folds inwards, so where the plasma membrane folds inwards, and that helps spread the electrical excitation much more quickly. And then we're going to go into these bands, and what we're going to do in a second is we're going to cut through like a little cut in these bands, and I'll go over this in a bit more detail at the end, but I really want to get into the sarcomere first. And we'll have a look at what a sarcomere is. So this is like one section we're cutting through. So we've cut through, and we're looking at the actual protein, so we're looking at a molecular level now within one of those muscle fibres seen in that muscle bundle of fibres. And there's two main things within one sarcomere. And there's lots of labels on these sarcomeres, but they're all down to the positioning of these two main things. And the first one is we have this thick band called myosin. So myosin is protein, and this is what's going to be used to do the muscle contraction. So that's the thick, darker band. And then you have the lighter band, which is referred to as actin. Now, next lesson, we're going to look at how this muscle contraction works. So if you're struggling to keep up, we're going to go back over this exact thing in more detail next lesson. But first of all, we need to learn the different parts. So if I just show this image, oops, sorry. If I just show this image, we can see the different parts. So the first thing to learn is you have an actin filament, and they're actually joined up here. And then you've got these myosin filaments, so they're both proteins. Now you need to learn what all these bands and names mean, so I'll try and go through them. So first of all, we have the I band. So the I band is the section where it is actin only, and it's referred to as the light band. Now the way that I remember this is light, the second letter is I know it's a bit crude, but Li, light band, is the I band. And in my head I just know that actin is thinner and smaller, so the light one makes sense that it's thinner and smaller, more light could pass through it. So the I band is when it's just the actin filament on its own. Okay, you've now got the A band. Now the way I remember that is, first of all, you remember one, you know the opposite one is the heavy, H-E-A. I know, again, it's very crude, but the heavy band, and that's the myosin, that's the thick band, and that's trying to match up these lines here. This light line is trying to match up with these light ones, and this dark purple piece trying to match up with these purple proteins in here. So you've got the A band, which is the same. Now you get lots of questions, because what happens is, imagine you're putting your two sets of fingers together, and then you slide your fingers in across each other, and they interlock into place. It works just like that. So we've got these light bands, and they all slide, they get pulled inwards, all in one go, and then they stretch out again when you stretch a muscle. So this A band, because that's the length of the myosin heads, they never actually move. What happens is they pull the actin across them. So the length of the A band will never change, but the length of the I band could change. And therefore the H zone, which is the zone between the actin, so this is this section here, the H zone or the H band, that's the area when a muscle contracts. If you interlock your fingers completely, there's no space there, so the H zone will essentially disappear, and we just end up with this M line, which is the central line. Maybe M for middle is a way to remember that, the middle line. And then that leaves then finally the last thing to learn is the Z line. And the reason why it's a Z line, it looks a bit like a zigzag Z. I'll find you another image shortly and show you it. There's like a zigzag between these actin filaments. They don't all sit in a straight line. So the first thing I want you to do is have a go now, pause the video, have a go at drawing this out, see if you can do it off by heart on your own. Have a go at that. So if I show you a nice clear image, it's the same idea. So you've got all these bands again, these I bands, you've got these A bands, which is the thick one, the H zones, the M line would go here, and then the Z line, as you can see, it zigzags because the actin zigzags between the myosin, so that's an easy way to remember that. So have a go at drawing something like this, try and label it fully, and then check yourself. So here, this image is trying to represent, so you can kind of get an overall look at it. You can see where these bands, the dark and the light bands shown. You can see now why that one of these bundles of myofibrils, so that's one kind of bundle of myofibril, you can see how they become striated because you have these light and dark bands that we saw earlier on the photomicrograph. And now to go back to our kind of original image, the only kind of other thing we haven't mentioned yet is this sarcoplasmic reticulum, which is the muscle version of the endoplasmic reticulum. If you think about the amount of protein that's here and the fact that they're now multi-nucleated, so there's one nucleus here but there's others, we have lots of nucleus, lots of sarcoplasmic reticulum, which is essentially endoplasmic reticulum, RER and SER, and that's useful because we're going to be making lots and lots of protein. So the many nucleus, the many sarcoplasmic reticulum all spread out over the myofibrils is useful because we're going to be making lots of protein. We're going to be repairing this all the time because muscle is built up and broken down all the time. Okay, right, let's have a look at some questions. So the first question here, I'm going to send you these rather than go through them in the video, so I'm going to send you these. So the first question here, which kind of works for us, so question 3. So just notice I'm sending you these questions that will work for today's lesson and next lesson. So start, when you get them today, start on question 3, and then you can do 1 and 2 and the rest of them after the next lesson, because the next bits we haven't done yet. So 3 types of muscle in a mammal, so we know mammalian muscle because we've studied that. A rabbit's body can contribute to the fight or flight response. So complete the table below by filling in the blank boxes. Pretty straightforward, smooth skeletal or cardiac muscle in there. And then now you need to apply, well, what's the action? How's that going to be useful in fight or flight, so the survival response? Okay. Next question. This is a really common kind of question that you're likely to get, is some kind of image, or they might give you a photomicrograph, so a real image of the sarcomeres. So the thick lines in the A band represent a protein, so name that protein. Okay, then I'm going to move on. So next question here is proteins in skeletal muscle myofibrils are arranged in groups to form a repeating pattern. As the muscle contracts, the pattern changes. Describe how the pattern within a myofibril changes, the skeletal muscle contracts. Now we haven't done muscle contraction yet, but you could have a go at maybe starting this question based on what we were talking about, my fingers sliding over each other, so you can kind of talk about that. I've skipped a question here, so just be aware on question 5 now. So the image below is transmission electron micrograph, so it's a cut through. So we've got some labels here, and then try to label that. I'll let you have a go yourself. Okay, so if you can send me your notes and send me your answers to these questions, I'll just review which questions I want you to do. So question 5 you can do, question 4A, and then question 3. Okay, thanks very much guys, see you next time.